everyone, and welcome to today's Protein Expression webinar. I am Cassie Saltman of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Dr. Marija Dramicanin, Head of Protein Production Facility, Walter and Eliza Hall Institute of Medical Research, and Dr. Roland Leathers, Senior Strategic Alliance Manager, Cell Biology, Thermo Fisher Scientific. Dr. Dramicanin, you may now begin your portion of the presentation.
Thank you very much. So my name is Marija Dramicanin, and I head a protein production facility at Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. And I am delighted to give you an overview of recombinant protein production for advancing medical research. So firstly, I would like to start by introducing my team and my laboratory. We specialize in recombinant protein production for a variety of downstream uses to help fast-track research at our institute. We offer a number of services, including expression in bacterial, insect, and mammalian cells, purification, and characterization. In these images, you can see my wonderful team. We are a team of 5 highly experienced protein biochemists. And you can see also our wonderful new and relatively new laboratory. So we are a facility established in 2021 in Melbourne, Australia. And in the first year of our operations, which was last year, 2022, we delivered over 100 projects for researchers at our institute. So now I would like to tell you a bit about recombinant proteins. What are they and what can we use them for? Recombinant proteins are proteins encoded by recombinant DNA that has been cloned in an expression vector that supports the expression of the gene and translation of messenger RNA into a protein. So recombinant proteins can be used for a number of different applications, and I just want to mention a few that are focus of our facility because of the research interest at our institute, and these include biochemical and cell-based assays for drug discovery, in vivo studies, discovery of therapeutic antibodies and antibodies, structural biology techniques like X-ray crystallography, electron microscopy, NMR, SPR, and other biophysical techniques. So when we receive requests for the protein production, the way that we start is by diving deep into the literature review. So what we aim to do is find out as much as we can about the protein that we are going to produce. And we also like to use available resources like UniProt. And as structural biologists, so this is the background that we have at the facility, we really like to look at the experimental structural biology data about our protein of interest. And if experimental data are not available, we also go for AlphaFold 2 and create a model of the protein structure, just like the one that is depicted in this figure presented here. So after we do a deep literature dive, we move on to a construct design. So this is part of the process that I want to emphasize is absolutely critical and the most important. And here I also want to emphasize that what you should be looking into is the downstream use of the recombinant protein. So we put a lot of emphasis. We're designing construct, having on our mind the downstream use of the protein that we are making. So first thing is a protein sequence. And when it comes to the protein sequence, for majority of the downstream uses, it is preferable to have a full-length protein, but this is not always possible, and this is due to various reasons. It can be a difficult to make membrane protein. Protein can have a lot of unstructured regions and be difficult to express and purify. So sometimes in those cases, we recommend looking into a folded domain for the expression, again, being informed by models and available experimental data. After we decide the protein expression, we look into a expression system. So choosing expression system is quite critical, and I'll speak about expression systems a little bit later in my presentation. We also recommend codon optimization of the gene for the expression suitable for the expression system. We also recommend choosing an appropriate vector for the expression system and also taking into consideration characteristics of the protein of interest. Again, it's really important to choose appropriate affinity tag suitable to the downstream use of the protein. Affinity tag can be part of the vector, or you can also add it to your protein sequence. Again, I want to speak a little bit more about the affinity tag. So affinity tag is important for the protein purification, and also it can be important for the downstream use. So in the cases like imaging, sometimes we will want to have a short tag, such as flag tag, that we can bind with the appropriate antibody and visualize our samples. But there are other downstream applications like crystallography, where it's preferable to have affinity tag removed. In these cases, we want to consider introduction of the protease cleavage site so that the affinity tag can be removed. And it is important to consider what type of protease we want to use, depending on, again, our protein of interest and final use. So when it comes to secreted proteins, it is important to include, obviously, signal peptide. And signal peptide can be part of the native protein sequence, or you can add one. Once we have a construct design, we reviewed, we are really happy with what we created, we can go into a synthetic gene ordering. So there are companies that offer this service, or one can do the cloning in the lab, depending on the preference. Also, it's important to consider that construct that you might want to use is already available on public domains like Adgene, or maybe somebody is willing to share them as part of a collaboration. So after the construct design, I would like to speak about the protein expression. So I want to focus on the 3 expression systems that we use at the facility. So first one, and speak about the advantages and disadvantages of each. So the first one is bacterial cell expression system. So this is normally what majority of protein biochemists will start with. And the reason is that this is quite a low-cost system. It is fast, but it has limitation. And the major one is that it doesn't provide post-translational modification. You can see here the simplified figure explaining the process of the bacterial cell expression. And in most case scenarios, it can include going from DNA to the product several days. So it is pretty fast. It includes transformation of the plasmid with the gene of interest into specialized bacterial cell lines. And there are different options here depending on what you aim to achieve. This is followed by the selection and expression test. If everything goes well, we go into the scale up, and finally purification. So that all looks great, but it doesn't, again, provide necessarily everything that we need. We often work with human proteins that require post-translational modification. And for these particular purposes, we have additional 2 expression systems. And the first one that I want to address is insect cell expression. So when it comes to characteristics of this system, it is in the range of the medium price. It allows post-translational modification, but the process is slow. Process takes a very long time. Again, I have here a simplified figure of the whole process, and you can already see that is a bit more complicated. So the first step includes recombinant BACMID DNA that is transformed together with the plasmid with your gene of interest into specialized bacterial cells. This is followed by selection process that includes blue-white colony selection. I'm not going to go into the details. After this comes the DNA isolation. And when we have a BACMID DNA with the gene of interest, we move on to the transfection of the insect cells. This is followed by virus amplification. And this can be anywhere from 1 to 3 different virus amplifications depending on the potency of the virus that you isolate. Virus amplification is followed by the expression test, and if that is successful, it goes into scale-up and purification. So the second system that we use when it comes to eukaryotic cells is mammalian cell expression. This system is normally of high costs, but it is fast, and it also provides cross-translational modifications. So initially, the process includes transformation of plasmid with the gene of interest into bacteria, selection, and DNA isolation. So the point of this is to generate enough of the DNA to do the transfection of the plasmid with the gene of interest into mammalian cells. This is then followed by the expression test, and if everything works all right, again, we go into a scale-up and purification. So these are the three expression systems, and we adapt them to different recombinant proteins. Once we express the protein, in all cases, the next step is purification. There are a number of ways to purify proteins, and this should be adapted to different recombinant proteins and their needs. Here, I just wanted to present typical three most used purification protocols. And for the purification, we use specialized equipment, FPLC or HPLC. Normally, as a first step of purification, we do affinity chromatography. So when it comes to affinity chromatography, this step is, of course, adapted to the affinity tag that we have in our construct. We use both loose resins or pre-packed columns. This, again, depends on the project. So this is the first step of isolating our protein of interest. But normally, this doesn't come out as pure as we would like to. So we proceed to a second most commonly used purification step, which is ion exchange chromatography. This purification step is basically based on the separation of the proteins based on the different protein charges. So this leads to further purification of the sample. And at our facility, in our labs, we like to finalize the protein purification with size exclusion chromatography. As name says, this process separates proteins based on size. But this is also protein purification steps that provides additional information. Looking at the size exclusion chromatogram, we can also conclude whether the sample contains aggregates, and also we can assess the quality of the sample in terms of solubility and also how homogeneous it is. We also use size exclusion chromatography to exchange buffer into a final buffer that is appropriate for the downstream use. We also put a huge emphasis on aliquoting and also having protein as concentration and aliquot size that is appropriate for our users. We also insist on flash freezing samples in liquid nitrogen and storing at -80. This, from our experience, is the best way to keep protein stable for longer periods of time. So apart from size exclusion that we use to assess the quality of our sample, we also have additional two techniques for the biophysical characterization at our facility, and these include size exclusion chromatography coupled with multi-angle light scattering, or in short, SECMALS. This is a very useful technique that we use to determine absolute molecular weight of protein samples. And here you can see a figure where we assessed the molecular weights of 3 different proteins. Of course, we have BSA as a standard control. This technique, apart from determining molecular weight, can be used to determine oligomeric state of the protein and also to study complex formations. Apart from SECMALS, the facility is a standard part of assessment of the quality of our sample. We use differential scanning fluorimetry, or DSF. Again, this is another example of a study of a protein depicted in this figure. So basically, the way that this technique is used to assess whether the protein is folded. And the technique means that we introduce the high temperature into our protein sample, and we follow unfolding of the protein through the intrinsic fluorescence that comes from tyrosines and tryptophans. And we come to a temperature of inflection, TI, which is telling us at what temperature the protein gets melted. So we can follow whether the protein sample is folded. We can also use this technique to study interactions of proteins with ligands. So these are the techniques that are available at the facility, but we also partner with our proteomics facility to do intact mass spec. We consider this to be extremely important quality control of the sample. We also have mass photometry available, and also techniques like SPR, ITC, and more. Of course, after we wrap up and we're happy with the quality of our sample, we hand over recombinant proteins to our researchers, and they do the functional studies and confirmation of the protein, which is, of course, the ultimately most important segment. So I've presented basically general overview now of process of expression, protein expression, purification, and characterization. But now I want to give you a bit more details about the two eukaryotic systems that we use at the facility, and why we like them, and give you some of the examples of the proteins that we make. So at the protein production facility at WEHI, we are using XP-SF9, high density insect cell expression system. These cells are derived from SF9 cells. We like to call them eco-friendly system, and I want to explain why. We at the facility grow 1 liter of these cells per 3-liter flask, and we infect the cells at high densities. From what we've seen with number of projects that we've conducted so far is that the reduction of waste and the yields of the proteins are just impressive, I have to say. We've seen examples where to achieve the same yields, we went from 20 flasks growing half a liter, of the cells, to just growing 1 liter of these high density insect cells to achieve higher or same or higher yields of the protein. So apart from that, this system is very robust and reliable. And what we like the most about it is that we can use it for both intracellular and for the secreted proteins. And I just want to give you some of the examples of the proteins that we made at the facility using this system. These are 3 different antigens. You can notice they have different profiles, reflecting complexity of producing different recombinant proteins. And apart from the high density insect cell expression system, we also have an alternative that is a mammalian XP293 expression system derived from HEC293 cells. Again, we see this as an eco-friendly system. We observed significant decreases in the waste, and this is, I think, the main reason why we like this system. It is, again, robust, reliable, and it is fast. We mainly use this system for the secreted proteins, although we did have some success with intracellular proteins, mostly on a lower scale, but this was still appropriate for techniques like cryo-electron microscopy, where a small amount of samples are sufficient. Again, here are several examples of the proteins that we made using this expression system. And here you can see we made antigens and different types of recombinant antibodies. We use variety of different sizes of the size exclusion chromatography columns. This is adapted to, again, the yields we try to achieve. You can see that they all look slightly different. I don't have time to get into the details of that. I just now want to move and talk. I think so far I've said a lot about what are the advantages of this system, but I want to also mention what are the challenges overall in recombinant protein production. I presented this beautiful picture by David Goodsell from the PDB because it shows experimentally solved structures of different proteins, so you can see how variable and how beautiful they are. And at the same time, I consider this to be the biggest challenge, but also the most interesting part of the job that we are doing. Sometimes I like to say there are as many challenges as proteins, but that's a bit too much potentially. So going through the challenges that I've listed here, these are some of them, is construct design and optimization. Again, I want to emphasize how important it is to really think carefully about the construct design, because if this is done properly, you don't have to go and redo everything. So it's quite critical. Optimization as well. The better the construct is designed, the less optimization is needed. Again, selection of appropriate host system. I hope that I've given you some indications of where you should go for one or the other. Expression and purification challenges, again, different expression levels, difficulty to purify the protein, etc. We do find that in most cases, if protein can be expressed to the sufficient level, we normally can optimize the purification, but then again, that's not always the case. Then protein folding and solubility. Here, I want to really emphasize how important it is to have those extra quality control. SDS page is great. Maybe you can see a fully pure band, but that doesn't mean anything when it comes to protein folding and solubility. You really need that size exclusion chromatography and additional QC measures that I've mentioned. You really want to see that. Again, scaling up protein production. There are a number of cases where the small scale works, but then you want to scale up to produce tens and hundreds of milligrams, and you see that it doesn't go that easy. Stability and storage issues. Again, each protein is stored for itself, and some are stable for months and years. Some have to be used in a really short amount of time. High production costs. This can be related to different expression system, and again, what are you after? What type of yields? Quality control and batch consistency. Again, I want to emphasize the importance of quality control. The better this is done, the more consistent batches are. So, always keep on mind that on your mind and do as many quality controls as you can. Last thing that I want to say is that I really feel like I have to mention the next best thing in protein biochemistry. So artificial intelligence and machine learning are taking over a lot of things, and it is part of the protein biochemistry revolution as well. So you can see this, very flashy nature and science covers, and I would argue that we live in the most exciting times for protein biochemistry. Myself and my lab are really interested in getting involved. So there have been significant advances in using AI and ML to, as they say, dream up brand new synthetic proteins for any uses. So I think that this is very exciting, and this is where I want to be, and something that we've been exploring. And I just want to finish with a picture of my team in these beautiful surroundings in Australia. And again, I'm happy to answer any questions you have for me. And if you want to find out more about us, you can visit our Institute webpage. And this is it for me. Now, I would like to hand over to the next speaker. Thank you.
Thank you, Marija. I would also like to say hello, ladies and gentlemen in our audience. On behalf of Thermo Fisher and myself, I would also like to wish you a warm welcome and thank you for choosing to spend time with us today. Following on from Marija's presentation, I would now like to look at how we can enhance protein expression efficiencies using the XP transient protein expression systems. Over the past decade, we've witnessed a strong interest in the ability to express human, mammalian, and viral proteins for research purposes, commercial purposes, and therapeutic purposes. This was particularly so during the recent COVID pandemic, where the demand for proteins carrying physiologically relevant human post-translational modifications increased massively as the world set about developing COVID vaccines and anti-COVID antibodies. And it is anticipated that the need to express physiologically relevant proteins rapidly will only increase as organizations such as CEPI, or the Coalition for Epidemic Preparedness Innovations, and the G7 group of countries have announced their vaccine moonshot plan to cut the time required for novel vaccine development to under 100 days. By their very nature, such programs will require protein expression systems allowing the rapid expression of significant quantities of physiologically relevant proteins. In this context, today I would like to introduce you to Thermo Fisher's XP systems, which are fully optimized and integrated transient expression systems that will allow the expression of gram per liter amounts of proteins in many cases. As we've seen during Marija's presentation already, there are numerous considerations when thinking about protein expression in one's own lab. And so I thought we would do here is just to have a quick look at some of the points one might need to consider when setting up a protein expression lab. These include understanding which proteins will need to be expressed in your facility. In particular, what is their natural origin and site of expression of these target proteins? Are these intracellular proteins or membrane-bound proteins? You'll also need to understand which applications these expressed proteins will be supporting. And we'll look at this further in the next slide. Which host systems will be required for the expression work? When we talk about the host systems, we're referring to the cell type that's used to express the desired protein. Some examples, as shown by Marija, would include bacterial expression systems, mammalian, insect, and yeast, and so on. In terms of the expression system, do you want to work with an optimized, commercially available expression system, or do you need to develop your own expression system? Although in today's presentation, we're essentially discussing transient expression of proteins, you may also need to understand whether a transient or a stable expression system will fit your needs best. And I'll go into this in a little more data in a later slide. Additional considerations will include an understanding of the throughput that will be required. How many proteins will the lab need to express in a given time? In terms of scale, how much protein will be required by the various projects requesting protein? Are we talking micrograms or grams of protein? How will you handle incoming requests, and how will they be prioritized? And importantly, what quality of protein will be required by the applications the proteins are to be used in? In this context, some further questions that might be of importance include, will the protein be used in a research environment or a clinical environment, as this will dramatically change the conditions under which the proteins are expressed? What purity of protein is required by the end user in their application? Is the activity of the protein important to the end user? Understanding these points will help direct you towards an appropriate expression system. I mentioned that an understanding of the applications will be used. I will mention that the understanding of the applications that will use the expressed protein will be important, and some applications are described in this slide and range from studying protein function to screening for drug candidates to understanding protein structures. Each of these applications may impose specific requirements on the type and quality of protein being expressed, and it's important to understand this. As we mentioned previously, producing a protein for research applications will be different to producing a protein for clinical use, as this will require establishing cGMP manufacturing processes and using qualified raw materials. One important question is certainly to understand whether post-translational modifications are important for the study in hand. In some applications, such as determining protein structure, it may actually be desirable to have a simplified PTM profile, or at least a more homogeneous post-translational modification profile to facilitate downstream analysis. Marija has already highlighted some of the challenges faced in the field of protein expression, and here are a few more that expression laboratories may have to consider, and which we at Thermo Fisher, as technology developers, are trying to address. As touched upon already, the first challenge is simply the variety of different protein classes that an expression lab has to deal with today. The second is the increasing number of demands to express what are generally considered to be challenging proteins, such as membrane-bound proteins. Thirdly, antibodies are no longer a single class of protein target, as there are now many different antibody formats, each of which may require special considerations. Finally, we see labs having to deal with an increasing number of demands for greater amounts of protein at higher quality. So how does an expression lab deal with all these challenges when there is no one-size-fits-all protein expression system to deal with protein expression requests. Well, this is really where the concept of having a protein expression toolbox comes in. The toolbox will contain a number of components which provide the researchers flexibility in trying to express proteins of different classes with varying degrees of post-translational modification in different amounts and for different applications. The toolbox will contain various expression vectors with regulatory elements, such as promoters and terminators, which are necessary for the protein expression and which have been tailored for specific hosts and applications. Expression vectors may include fusion tags, which can be expressed as fusions with the target protein to aid in purification, detection, or localization. Some vectors may allow the expression of proteins in an inducible manner to allow researchers to control when protein expression occurs. And of course, the toolbox will contain various expression hosts, as these are the workhorse cells that will be used as protein expression factories. And when we talk about expression hosts, this slide shows the 5 major host systems used for protein expression today, ranging from cell-free expression systems to bacterial systems, all the way through to yeast, insect, and mammalian cell hosts. As we already touched upon, one of the first considerations in any protein expression project is the host expression system. So what are the main considerations here? Well, there are a number of parameters that may need to be considered, but a few of the basic considerations include, does the project require post-translational modifications or not? How easy does the expression system need to be to use? How simple or complex are the culture conditions involved? And what yields of protein are required? As mentioned during my introduction, the past decade has witnessed a strong interest in the ability to express human, mammalian, and viral proteins using what were considered to be harder to work with mammalian expression hosts. In considering expression in mammalian hosts, historically, if you wanted to express moderate amounts of protein in a mammalian expression system, you would turn typically to making a stable cell line. But as we know, this process can take many months to a year, which is a long time to wait for your protein of interest. To try and shortcut stable cell line generations, groups turned to transient expression systems, which were built from existing components derived from various sources that had never actually been optimized for this purpose. The net result is that one could use these do-it-yourself transient systems to express proteins rapidly, but typically the yield of protein would usually be quite low. So given this bottleneck, Thermo Fisher set itself a research goal to develop transient expression systems that would allow the expression of protein yields typically associated with stable cell lines, and to obtain these yields in days rather than months. So how are we going to do this? Well, internally at Thermo, through the efforts of past product development programs, we've established considerable expertise in developing media systems, transfection reagents, cell line selection capabilities, and vector design, which would serve as the foundation for this new coordinated research endeavor. Some of the system design constraints that we have posed upon ourselves are shown in the orange bar. The systems should provide highly reproducible protein expression yields. They should provide ease of use and should be easily scalable for both larger scale as well as smaller scale expression needs. If we look at the bottom half of the slide, we see the components which are common to most transient protein expression systems, including the producer cells, which are the real workhorses of the entire system, the culture medium used to expand the host cells and produce proteins, potential enhancer reagents, transfection reagents, and vectors. And we use design of experiments, or DOE approaches, to optimize each component individually, and then use DOE again to optimize the entire system. The output of this development program has so far resulted in 3 XP transient protein expressions systems shown on this slide. The XP293 system, which is used extensively as a biologic screening and discovery tool, and also in structural biology. The XB-CHO system, used extensively as a biologics development tool. Remember, some 60% of biologics are manufactured in CHO cells today, and that's why CHO is considered the gold standard expression platform in the biologics industry. XB-SF, used notably to express intercellular proteins, multimeric complexes, and viral vectors such as AAV, which are used in gene therapy applications today. These are all fully integrated and optimized expression systems. The XB293 and XB-CHO systems use mammalian hosts, while the XB-SF system uses an insect cell host. And so what I'd like to do now is to provide you with a little bit more detail on each of these transient protein expression systems, starting with the XB293 system. The XB293 system allows transient protein expression in suspension-cultured HEC293F cells. The system allows expression of gram per liter amount of protein in roughly 7 days. This fully integrated system comprises high-density and high-yielding 293 cells adapted to suspension culture, optimized culture media, and accompanying reagents. More recently, the XB293 system was complemented with additional XB293 cell lines and reagents for specific applications, such as structural biology and inducible protein expression. These can be seen in the bottom right-hand corner of the slide. In the GNT1- version of the XB293 cell line, we have knocked out the N-acetyl glucosylamyl transferase 1 gene, thereby preventing the formation of N-glycans. This dramatically simplifies the PTM profile of the expressed protein, thereby facilitating structural analysis of the protein in question. The XB293 inducible cell line allows for inducible and regulated expression of the target protein and might be useful, for example, when expressing toxic proteins which might otherwise kill the producing host cell. The XB293 inducible GNT1- cell line allows for inducible and regulated expression of proteins with a simplified PTM profile. The other methionine deficient reagents shown here are for performing methionine labeling of proteins to be used in X-ray crystallography and NMR studies. The XB-CHO-S system allows transient protein expression in suspension-cultured CHO-S cells, and we commonly see expression yields of up to 3 grams per liter. Although stable CHO lines are routinely used in the industry to manufacture proteins at high yield, the yields described here for the XB-CHO system were previously unheard of in a transient CHO system. One of the consequences of this groundbreaking performance is that biologics groups can now start their development projects in a high-yielding transient CHO system rather than switching to another transient host, such as HEC293, to obtain the high yields required, which was previously the case. The XB-CHO cell line is also available as a GMP banked cell line, along with complete audit trail and documentation package for the submission to the regulator, should you wish to produce protein for clinical use. In terms of the system components, this is a fully integrated system comprising a high-density and high-yielding CHO-S cell line adapted to high-density cell suspension culture, optimized culture media, and accompanying reagents and feeds, while the XB-stable production medium provides a path for transitioning your transient workflow in XB-CHO to a stable expression context, should that be required. In this slide, we're looking at overviews of the recommended expression protocols for both the XB293 system at the top of the slide and the XB-CHO system at the bottom of the slide. If we first look at the general protocol for expressing proteins transiently in the XB293 system, we see that on day 1, prior to transfection, our high-density log-phase XB293 cells are subcultured to a density of about 2.5 to 3 million cells per mil. On day 0, the day of transfection, the cell density is first adjusted to 3 million cells per mil in fresh culture medium, and then transfected with the diluted XB293 transfection reagent complexed with the plasmid encoding the target to be expressed. 1 day post-transfection, we add our XB293 expression enhancers to the culture to boost expression. Proteins can then be harvested between day 5 and day 7 post-transfection. For the XB-CHO system, shown in the bottom half of the slide, there are a number of protocol options depending upon how much protein is desired and how quickly it is needed. For the 3 protocols options shown here, referred to as standard, high, and max titer protocols on the slide, the start of the process is the same. 1 day prior to transfection, the high-density log phase XB-CHO cells are subcultured to a seeding density of 3.5 to 4 million cells per mil. On day 0, the day of transfection, the cell density is adjusted to 6 million cells per mil in fresh culture medium, and then transfected with the diluted XB-vectamin-CHO transfection reagent, complexed with the plasmid encoding the target to be expressed. The expression enhancer and the first feed is added 24 hours post-transfection. In the standard titer protocol, protein is harvested 8 to 10 days post-transfection. In the high titer protocol, after the addition of the enhancer and the first feed, cultures undergo a temperature shift by returning them to an incubator set to 32 degrees instead of the regular culture expansion temperature of 37. This temperature shift provides a means to boost expression yields. Protein is then harvested 10 to 12 days post-transfection. Finally, the max titer protocol, after the addition of the enhancer and first feed, the cultures undergo a temperature shift by returning them to an incubator set at 32 degrees. Then on day 5, a second feed is given to the cells. Protein is harvested 12 to 14 days post-transfection. As you can see from the experimental schematic, each protocol is associated with a shorter or longer expression run, but as you will see on the next slide, the protocol also has a major impact on the yield of proteins achieved. In this slide, we're looking at the kinetics and yields of protein achieved for a human IgG target expressed in both the XB293 and the XB-CHO system. The brown line indicates that a maximum yield of approximately 1 gram of protein per liter was achieved after 7 days in the XB293 system. The 3 blue lines indicate the yield of protein achieved in the XB-CHO system using the 3 different expression protocols described in the last slide. The standard titer protocol resulted in approximately 1 gram per liter after 8 to 10 days. The high titer protocol, which differed from the standard protocol by using a temperature shift, resulted in approximately 2 grams per liter after 10 to 12 days, while the max titer protocol, which differed from the standard protocol by using a temperature shift and a second feed, resulted in approximately 3 grams per liter after 12 to 14 days. So again, depending upon how much protein is needed and how quickly you need it, one can select the appropriate XB-CHO protocol. So how do these yields compare with classical transient HEC293 and CHO expression systems? Well, this table provides an idea of the expression efficiency of the fully integrated and optimized XB systems compared with the standard adherent transient expression systems. Firstly, for the HEC293 system, if we compare the expression of a few different protein targets in a standard transient 293 system, the middle orange column, with the XB293 system, the left-hand blue column, we see significant increases in expression yield in the XB293 system for those targets. If we now compare the expression of a few different target proteins in a standard transient CHO system, the right-hand orange column, with the XB-CHO system, the central blue column, we see a massive increase in expression yield in the XB-CHO system. And it's this expression difference which really has been a game changer for the biologics industry in particular. The right-hand blue column also shows that generally As a general rule of thumb, the XB-CHO system results in higher protein yields than the XB293 system. However, it's important to remember that if we were to review the expression of a broader range of targets, we would actually see that not all proteins can be expressed efficiently in XB-CHO. So having access to a second host system, such as the XB293 system, is a very useful component of any expression toolbox. Unfortunately, I don't really have time to go into much detail, further detail, but as mentioned in earlier slide, the expanded XB system toolbox includes various engineered cell lines and reagents which are suited for specific applications such as structural biology and induced expression. This also includes GMP-banked XB293 and GMP-banked XB-CHO cells for manufacturing in a GMP environment. In the last part of this presentation, I'd like to turn our attention to protein expression using insect cells. So let's start with the question, why express proteins in insect cells? Marija has already touched on this, but I will also say that insect cells offer several advantages over other hosts. They provide high levels of protein expression with post-translational modifications approaching that of mammalian cells. They provide an easy route to scaling up expression. They require only simple culture conditions. Indeed, a simple culture environment of 27 degrees with no CO2 control is sufficient. And insect cells are generally readily adapted to high-density suspension culture for large-scale expression. With that, I'd now like to provide a brief introduction to one more XB system, namely the XB-SF system, which completes our range of XB transient protein expression systems by adding an SF9 insect cell host to the toolbox. The XB-SF system allows transient protein expression in suspension cultured SF9 cells. The system represents a big step up in expression technologies because when its performance is compared with that of other commercially available insect expression systems, we routinely see up to threefold higher protein yields. And maybe more importantly, the time required to produce the baculovirus encoding the transgene is significantly reduced. Indeed, this is the first expression system to use both fully chemically defined culture media and an expression enhancer, which together provide consistent growth and expression reproducibility. Typically, the time required from baculovirus production to protein expression is just 6 to 10 days in the XB-SF system. And in terms of the system components, this fully integrated system comprises a high-yielding SF9 line adapted to high-density cell suspension culture, optimized culture media and accompanying reagents, as well as the back-to-back vector system and DH10 back competent cells, allowing generation of the recombinant backmid DNA for virus production. In this slide, I would like to compare the time to protein using a classical insect expression system shown in the top half of the slide with that of the XB-SF system shown in the bottom half. Looking first at the classical expression systems, the infective baculovirus is produced from backmid DNA using an adherent insect cell culture, resulting in the production of only a nominal amount of P0 virus. As the amount of virus produced is so small, typically several rounds of virus amplification may be required to produce sufficient virus to allow the expression of the desired protein in insect cells. The two major problems with this are that each round of amplification takes time and the viral quality will be degraded with each round of amplification. The net result is that the classical protocol will typically take some 12 to 20 days to generate protein when starting with backmid DNA. In the XB-SF system, which uses high-density SF9 cells in a suspension culture, it's possible to generate significant amounts of P0 virus without any further viral amplification steps. This reduces the time to protein from backmid DNA to just 6 to 10 days and ensures using only the highest quality viral stocks by using the P0 virus for infection. So let's take a quick look at the XBSF9 protocol in a little more detail. In the top half of the slide, we're looking at the first step in the process, which is the generation of the baculovirus by transfection of the BACMID DNA into our XBSF9 cells. So to make virus on day one, insect cells in log growth phase are diluted to the appropriate density for transfection. The BACMID DNA encoding the gene of interest is then complexed with the diluted transfection reagent and transfected into the insect cells. 4 to 5 days post-transfection, the P0 virus can be harvested and either used directly to express protein following titration, or the virus can be stored in the dark at 4 degrees centigrade for several months for future use. In the bottom half of the slide, we're looking at an overview of the second step in the process, which is using the baculovirus to express the desired protein. Here we see that on day 5 of our log phase insect cell suspensions are diluted to the appropriate seeding density, which corresponds to approximately 5 million cells per mil, and the XBSF enhancer is added to the cells. The role of the enhancer is to boost viral infection when the baculovirus is added to the cultures the next day. On day 6, the insect cell suspensions will have attained a density of approximately 7 million cells per mil, and the titered baculovirus can be used to infect the cells. Proteins are typically harvested from the cultures at time points between 7 to 10 days, and as we'll see in the next slide, optimizing the protein harvest time may be beneficial. So here I'd like to share some example expression data using the XBSF system, in which we're looking at the expression of a challenging target known as CB2. CB2, otherwise known as the cannabinoid receptor, is a membrane-bound GPCR. In this example, following an expression and harvest time course study shown in the bottom center of the slide, 48 hours post-transfection was identified as the optimal protein harvest time point. For this specific target protein, beyond the 48-hour time point, protein quality decreased as cell viability declined. In terms of protein yield, we see from data shown in the bottom right-hand corner that when compared with a classical SF9 expression system, the XBSF system achieved a 10-fold yield increase, which of course represents a significant improvement in protein yield. So to summarize this part of the presentation, I've introduced you to our 3 host systems, namely the XBSF, XB293, and XBCHO systems. And together with the expanded XB toolbox comprising the various engineered cell lines and reagents for specific applications, we believe this provides you with a highly functional and efficient toolbox for expressing proteins of multiple classes in your own lab. In addition, I would like to mention that you can also access these expression systems through our service functions. We use all 3 expression systems to provide protein expression services at the request of our clients. If this could be of interest, then please just inquire, and we'll be happy to share further details. I would also like to mention that protein expression per se is just one component of the entire expression workflow. At Thermo Fisher, we call this expanded workflow the cloning, expression, purification, and analysis, or CEPA workflow, which is depicted here in steps 1 to 5. Now, there's far too much detail on this slide to go into today, other than to say Thermo Fisher is here to provide you with a seamless and integrated solution across the entire CEPA workflow, helping you to simplify the task of producing the right amount of purified protein at the right quality for your application. I'd also like to take the opportunity to mention that Thermo Fisher has a strong history of development in the field of protein expression. Indeed, the first expression XB systems were launched over 10 years ago, back in 2012. And since then, the XBCHO and XBSF systems were launched in 2015 and 2018, respectively. During this time, all 3 expression systems have been cited extensively in the literature, and we have now made these citations available to you through our online publication hub, which you can find online. And finally, should you have questions about your new protein expression project, I encourage you to reach out to Thermo Fisher's global network of field application scientists who will be happy to support you in executing your projects. And with that, I would like to say a big thank you to our guest speaker, Dr. Marija Dramicanin from the WEHI Institute in Melbourne, and to you, our audience, for listening to today's presentation. And I will now hand the presentation back to our host, Kathy, for the Q&A session.
Thank you, Dr. Dramicanin and Dr. Leathers, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question is for Dr. Leathers. Which vector do you recommend using with the XB293 and XB-CHO mammalian expression systems?
Yeah, thank you for the question. I think the simple answer is that any vector you have used previously for expression in a mammalian host can be used in the XB293 and XB-CHO systems. However, for highest expression yields in both the XB293 and XB-CHO systems, we recommend using the pcDNA 3.4 topo vector. This vector contains the native full-length CMV promoter and a WPRE. That's a woodchuck post-transcriptional regulatory element downstream of the cloning site. Both of these elements contribute to high-level gene expression. Expression yields with the pcDNA 3.4 topo vector are generally 2 to 3-fold higher than with the alternative pcDNA 3.3 topo vector, which does not carry a WPRE sequence. However, higher yields from the pcDNA 3.3 vector are still 2 to 5-fold higher than working with a standard pcDNA vector. But of course, I think one should remember that expression yields do not only depend on the vector, but also on the protein target itself.
Fantastic, thank you. All right, our next question here, it looks like, is for Dr. Dramicanin. What recommendations do you have for setting up a new recombinant protein expression lab, including choosing the expression system and addressing challenges when producing proteins for the first time?
For this question. So I just want to say that I think that this was really nicely covered by Roland. And what I want to emphasize in this case is to choose the expression system, vector, and host organism or expression systems that best match the protein characteristics and desired applications in order to maximize the chances of the successful protein production.
Great, thank you. It looks like we have another question for Dr. Leathers. This question is, if I decide to work with the XB293 and or XB-CHO systems, how do I know that the system has been set up correctly and is performing optimally in my lab?
Thanks, Kathy. That's a great question because there's really no point establishing a high-performance expression system in your lab and not having the system work optimally. It's like driving a sports car in first gear all the time, and that just really doesn't make sense. To this end, we offer a positive control vector encoding a rabbit IgG, which serves as a technical control during your system setup and during any subsequent expression runs by allowing you to monitor your transfection and expression. Then, by comparing your expression yields achieved with the control construct, it's a straightforward process to understand if you have the system operating optimally in your lab. Should you see suboptimal results, our protein expression field application scientific team will be happy to work with you to optimize expression performance.
Great, thank you. And it looks like we have time for just one more question here today, and that question is for Dr. Dramicanin. The The question states, what are the key considerations and important factors to pay attention to when conducting eukaryotic protein expression experiments?
All right, so thank you for this question. I think similarly to my previous answer, I think the important considerations are again the vector, and it's really important to do appropriate construct design. Again, thinking of the protein characteristics, post-translational modifications, this is really important, and desired final application of the protein. I think that this is all the things that we have to take into consideration. Also thinking of eukaryotic system, we have to think of the time required from the DNA construct to the final product, and of course the yields that we are trying to achieve. So I think that Roland has covered this beautifully, and we know that there are different expression systems available there to suit different needs. I think that I want to end this answer and actually, talking about the recombinant protein expression and purification, just emphasizing how hard it is to work with different recombinant proteins, because I feel like they all have their specific needs. And not only that, again, the downstream use have their specific needs. So I think that this is really important thing to consider. And the more streamlined our procedures can be, the better and the easier. And, I just want to add that this is why I find high density expression systems to be working really well, because it's helping us with this complexity. So thank you. This is where I want to finish.
Thank you again, Dr. Dramicanin and Dr. Leathers, for your time today and your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.